Hello there everyone, and welcome to this Biology Mind A-Level tutorial on tests for carbohydrates. In the last lesson we learned that a 3D protein can have one of two structures, a globular structure or a fibrous structure. So globular proteins are more suited to metabolic roles, whilst fibrous proteins usually have a structural function. Now today we're going to do a bit of a topic flip and go along to think about how we test for carbohydrates. So we're going to look at Benedict's test, we're going to look how that's applied to real life in reagent test strips. We'll then go on to have a look at the iodine test, and then again look at how these are thought about and adapted into our everyday living life in colorimetry and biosensors. Let's have a think first of all at what type of sugars there are. Now there are two types of sugars, reducing sugars or non-reducing sugars. Now have a pause of the video and have a think from your chemistry knowledge what you think reducing sugars do and what you think non-reducing sugars do. Okay, so I'll reveal the answers. So reducing sugars do exactly as the word says, they are reducing agents. Now what are reducing agents? Reducing agents donate electrons. That is what defines a reducing agent. They reduce another substance. Now, I'm sure you can guess what non-reducing sugars are. They don't reduce agents. And they include most disaccharides, such as sucrose. Okay, so now that you know what a reducing sugar is and a non-reducing sugar is, I'm going to explain Benedict's solution and the Benedict's test. Now, I'm going to break it down into a little bit more detail because I think that makes it make a bit more sense. So try to follow me here and hopefully this will make this test make sense to you. So Benedict's solution is essentially copper sulfate. Okay, and I promise you this is relevant. So it's essentially copper sulfate. Okay, so if we want to test for our reducing sugar, we add Benedict's solution, which is essentially this copper sulfate. We then heat the mixture and we then observe the colour change. Now, before I go on to tell you what happens on the next slide, let's just have a think about this process. So we have our copper sulfate and we're adding a reducing sugar to it. So let me write copper sulfate here. So this is our Benedict solution. And if we have copper, so these are the copper ions within copper sulfate, within Benedict's solution. And if we have a reducing sugar, we're going to reduce our copper sulfate. Now, what does copper 2+ get reduced to? Remember, what do reducing sugars do? They donate an electron. So if we donate an electron to copper 2+, we're going to get copper 1+, or copper oxide. Now, this forms a coloured precipitate, okay? This actually forms an orange-red precipitate. So we'll look at that on the next slide. Oh, might be on the slide after that. So Benedict's test is semi-quantitative. Now, I've already told you that a precipitate forms if a reducing sugar is present, that orange-red precipitate. So all it means by saying that Benedict's test is semi-quantitative is that depending on how strong the orange-red solution is, this can be measured and this correlates to the concentration of reducing sugar in the solution. So Benedict's test doesn't only tell you whether reducing sugar is present or not in the solution, it actually tells you how high the concentration, roughly an estimate of it, of reducing sugar there is in this solution. So for multiple samples, Benedict's test should be carried out under standardised conditions, as I'm sure you're all familiar with, having done lots of experiments over the years. So this means ensuring all solutions are the same volume, so for example adding the same volume of Benedict's reagent to each sample, that they're given the same amount of time from when Benedict's solution was added to inspecting for the colour change. So you want to control these variables. We can then estimate which solutions contain the greater concentration of reducing sugar in comparison to the other. So let's just go on to the next slide. So this is what I wanted to show you. So remember what we're doing is our Benedict's solution contains these copper 2 plus ions. Now, copper 2 plus ions are reduced by our reducing sugar, if it's present, to copper, oops, it's refusing to write, to copper 1 plus ions, okay? And our copper 1 plus ions are an orange-red, they form an orange-red precipitate. So this means that the more copper 1 plus ions we form by reducing more of the copper 2 plus in Benedict's solution, the more orange-red precipitate we form. So from that, we can get that the more reducing sugar that there is in the sample, the more orange-red precipitate that we form. So here you can see that with increasing reducing sugar concentrate, we form a more orange-red solution. So blue would be the colour when there is no reducing sugar in the sample. If there's a tiny bit of reducing sugar in the sample, you'll get something in the green to yellow spectrum. And if there's lots and lots of reducing sugar in the sample, then we get the orange to brick red spectrum. Now remember, that's because we're reducing with our reducing sugar Benedict solution to this copper plus ions, which form this orange-red precipitate. So in the same way, we can actually use Benedict's test for non-reducing sugars, and you'll be thinking, what's she on about? You need the reducing sugars to reduce those copper 2 plus ions to copper 1 plus ions. Now, what we can do is if we add the non-reducing sugars, so if I go back to this slide here, where we had the Benedict's test for reducing sugars. Now let's say we add Benedict's solution to a non-reducing sugar. What's it going to do? Well, pause the video at this point and have a think. So if I add a non-reducing sugar to Benedict's solution, it's going to remain in this blue range. It's going to show that there's no reducing sugar present, only non-reducing sugar or something else. Now that's not good enough a test for whether a non-reducing sugar is present, because the absence of reducing sugar doesn't tell you that non-reducing sugar is present. It can be anything. So what we want to do is we want to do a specific test for non-reducing sugars. So we've got our negative Benedict's test already, having added the non-reducing sugar to the Benedict solution. So we've got our negative test. Now we're going to think, okay, is this a non-reducing sugar or not? And to do that, we've got to add dilute hydrochloric acid. Now, have a pause of the video and have a think about what you think dilute hydrochloric acid will do. Okay, so I'll tell you at this point. So remember, most of our non-reducing sugars are disaccharides. So if I draw out a disaccharide, let's say this is our disaccharide. Now, if I add hydrochloric acid and heat to that, I'm going to break it down into its monomers. I'm going to break it down into its monomers here. So, for example, if this here is a disaccharide, such as sucrose, I would break it down into its monomers, such as glucose and fructose. And these monomers are now reducing sugars. So that means that if I now add Benedict's solution to this, I'm going to have a positive result because I've got my reducing sugars present. Whereas if I add a Benedict's solution to this is not a reducing sugar. This is a non-reducing sugar. Whereas these are reducing sugars. So we need this hydrochloric acid to break down our non-reducing sugar to our reducing sugars. Now, bear in mind, this is acid we're adding to our solution, so we're going to want to neutralise it. And we just do that by adding some sodium hydrogen carbonate, for example, or it could be another sort of alkaline neutralising solution. So we have our neutralised solution. We're then going to do this exact same thing that we did before, because we've now essentially got our reducing sugars in our mixture. So we're going to heat the mixture and we are going to observe the colour change. And remember, with the colour change, what are we going to get? We're going to get an orange-red precipitate because we're going to get our copper plus, 2+ ions of our Benedict solution being reduced by our reducing sugars to our copper plus ions, which are going to form this orange-red precipitate. So I think a good way to summarise this is to have a think about what happens with reducing sugars versus non-reducing sugars. So I'll just summarise this here for you. So with reducing sugars, we're going to get a positive Benedict's test. So I'll write a positive here. Well, why don't we do that in like an orangey reddy colour, as close to orangey reddy as we can get. All right, now with non-reducing sugars, we're going to get a negative Benedict's test, but if we break our non-reducing sugar down with hydrochloric acid and then add Benedict's solution, we're going to get a positive Benedict's test. All right, so that's quite a good way of making a little summary of what we've learned there. All right, so that was quite heavy stuff. So now let's think about how this is applied to real life. So in daily practice, the presence of reducing sugars are efficiently tested for with reagent test strips. And these test strips contain a reagent which interacts with the reducing sugar when immersed in the test sample. And examples include urine test strips, which can test for presence of glucose in urine. And this occurs in something like diabetes, for example, where you get the glucose in the urine. And in the same way, you just compare the colour on the strip with the chart on the bottle. So the degree of colour change that occurs in the reagent can indicate the concentration of that glucose in the urine. And we can compare, as I said, this final colour on the test strip to the correlating concentrations on the bottle here. All right, so now we've talked about our carbohydrate tests. We're now going to talk about our iodine test for starch. So let's have a think about this iodine test for starch then. So iodine test for starch is also known as, the iodine solution is also known as Lugol solution. It's a brownish orange in colour and is formed by adding potassium iodide to water. So we get this iodine solution. Now, if starch is present, we'll get a dark blue-black colour. So that is your key takeaway message of this iodine test. If starch is present, we get a dark blue-black colour. All right, so now we've had to think about how we test for reducing and non-reducing sugars. We've had to think about how we test for starch. So now let's think about how we can quantify this. Now, all that means is getting a strong, hard value about how much of this starch or reducing sugar or non-reducing sugar is present in our sample. So something that we can use is colorimetry. So to get exact values of biological molecules, we have this method. And it's a quantitative method, and it allows this concentration of compounds to be directly measured. So it involves the use of this meter called a colorimeter, and it's a device that detects how much light is absorbed by the solution. The more concentrated the solution, the more light it will absorb. So how do we then correlate the amount of light absorbed to the concentration of the solution? Well, if we get 5 known concentrations of glucose solution, so let's say we know the concentration of 100 milligrams per litre of glucose solution, and we put that through the colorimeter and we get an absorbance of 0.5. And then we do the same for 200 milligrams per litre and we get the absorbance of 1. And so on for the various concentrations, so you can see them plotted here. We can then draw a line of best fit through all of those plots. And from that, we get a nice trend where we can essentially read off the graph. Whatever absorbance we have from just above 0 to 2.5, we can read off this graph what the corresponding concentration of glucose is. So that's how you use a calibration curve. And you might be given something like this in an exam, so familiarise yourself with what we just talked about there. Okay, so this is essentially what I've just described, but just in writing. So you find out what the colorimeter says and you put that onto the graph and you read off your line of best fit and determine at what glucose concentration your result would occur. Now, another way of quantifying how much of something is present in a solution is a biosensor. I don't know if any of you have ever seen any of this. So this is a monitor to detect how much glucose is present in a blood sample. And this is what diabetic patients often use. So all it involves is pricking the finger for some blood and putting that blood sample into the biosensor. Now, biosensors are devices that analyse the concentration of substances in a solution. And they do this in a different way to the colorimeter. So they contain a biological agent which interacts with the test substance. So examples include enzymes, antibodies, nucleic acid receptors, and cellular organelles. So the biological agent and the test substance react to produce a chemical signal. The chemical signal is received by the transducer, and the transducer converts this to an electrical signal. And this electrical signal can then be analysed to quantify the concentration of substance present in the sample. So that's how those glucose monitors work, which is quite interesting. So glucose biosensors contain the enzyme glucose oxidase. It oxidises that glucose on the blood sample. The reaction produces a charge that's turned into an electrical signal. Remember, we said the chemical signal is translated into the electrical signal. And this occurs by transducers in the machinery. And then the electrical signal can then be used to determine the value of glucose concentration in the sample. Simple. Okay, so that's us done for today. This was quite a long tutorial, nearly 20 minutes we've been here. But I hope that made sense, and if you think you need to recap any of those explanations there, then please do rewind the video and go back to have a look. But well done for following through, and I'll see you for the next session.